Hello everybody. In this presentation, I will be giving a simple introduction to amino acids and proteins. So I'll start with describing the basic chemical structure of an amino acid. You have a central carbon atom which has 4 valencies. The first is an amino group. The second is an acid group, the carboxylic acid. Here you have a hydrogen, and the fourth is an R group, which is the identity group of any given amino acid that imparts specific properties to that particular amino acid. The simplest amino acid that I'm going to draw out here is glycine. To draw any amino acid, you need to make a carbon which has these 4 different bonds. Go ahead and make these the amino group, the acid group making the amino acid, and in case of glycine, the R group is a simple H. So this is the simplest possible amino acid that you can have. Each of these amino acids could either be denoted by a 3-letter code, in this case gly, and a 1-letter code, which is the first letter of the name of the amino acid, unless you have other amino acids which are also starting with similar names, such as glutamic acid, which has a 1-letter code that's of E. The second simple amino acid that you have has a methyl R group. Now, as you can see that the carbon here is chiral, and based on the handedness, that whether it's left-handed, it's either an L-alanine or a D-alanine. These are known as stereoisomers. The next important amino acid that I'm going to talk about is proline. Proline sometimes is also called as an amino acid because the nitrogen of the amino group here is linked to a cyclic carbon group, and this is what makes these amino acid a rigid amino acid in terms of the structure. Another important amino acid that's found in the proteins is cysteine. The cysteine here you can see has a CH2SH group. This CH2SH group from 2 different cysteines can undergo oxidation reaction to form a disulfide bond, and this molecule is called as cysteine. These disulfide bonds are very prevalent in nature, and they are biologically very interesting to study. One famous example is that of hair straightening, where most of the chemicals or the heat treatment that you use are responsible for breakage of these disulfide bond, converting curly hair into straight hair. So this is a typical table that you will find in any biochemistry textbook, where the amino acids are grouped into different subcategories based on the nature of their R group. You can have non-polar aliphatic side chains, you can have aromatic side chains, then you can have polar side chains, which can either be uncharged, positively charged, or negatively charged. As I mentioned before, these different amino acids are either called by a 3-letter code or by a 1-letter code. In most cases, it's easy to memorize, except for cases like phenylalanine, tryptophan, asparagine, glutamine, aspartate, glutamate, etc. The smallest possible amino acid, glycine, has the least molecular weight, whereas the bulkiest amino acid is tryptophan, having a molecular weight of 204. The important property that I want to talk about in this slide are the different titratable groups that are present in these amino acids. The 2 common groups are COOH and NH3 that have similar pKa values across all different amino acids. What I mean by titration here is that if in a solution of amino acid you add OH equivalents, that is if you're adding a base, the first proton to be lost is from the carboxylic group, which gives you the pKa1 value here as shown in the graph below. The second group to be lost is from the amino group that gives you the pK2 value. Now here you can see that the net charge on this molecule changes from a +1 to 0 to -1. So from here you can define the isoelectric point for this particular amino acid. Isoelectric point is nothing but the pH at which your net charge on the molecule becomes 0. This is what is listed over here. What you can see here is that for most of these amino acids, the carboxylic group has a pK value of about 2, whereas for the amino group there's a pK value of about 9, except for cysteine here, which has a very high pK value for the amino group, and that is mainly because of the sulfhydryl group that it has in its side chain. Now the third titratable group that can be present in some of these amino acids, that is particularly the polar amino acid, is your R group, and based on whether it's positively charged or negatively charged, the pK value of the group can either be greater than that of the amino group, or it can be lower than that of the amino group. The amino acids for which the R group has a pK value greater than that of the amino group, it basically means that when you add the when you titrate it with an acid, the group that loses the proton first is going to be the R group in case of negatively charged amino acids, whereas it's going to be the amino group in case of the positively charged amino acids. So the order of loss of protons dictates the pK value of these amino acids, although I must mention that when these amino acids are present in context of a chain, that is in context of a protein, the pK values of the side chain could vary from what it is when these amino acids are isolated singly. So moving on from the amino acids to polypeptides and proteins. Now these amino acids, when they react with each other, a decondensation reaction, that is a loss of water molecule, can lead to the formation of a peptide bond, which is a CONH bond. Because of the bond being called as a peptide bond, when many of these amino acids come together, the condensation reaction happens, peptide bonds are formed, you get what's called as a polypeptide. The longer the polypeptide, the name then changes to proteins. The simple linear sequence of these linear sequence of amino acids in a polypeptide chain forms the primary structure of a protein. You can imagine the primary structure to be like a thread. Now these thread-like structures could then form a higher level of organization, which could be either spiral, which is called as an alpha helix, or it could be in form of sheets, which are called as beta sheets. These different secondary structures could interact with one another. That is, you could have a beta sheet that's usually denoted like an arrow, followed by an helix, followed by a random loop, and then again followed by a beta sheet to form what's called a tertiary structure. In most of the cases of the proteins, this level of structure is the structure that's functional within the cells. So this is called as the final folded form of the protein. And this folded form is the one that performs the function. In case of certain proteins, you could have individual units, which is depicted here in the form of a tertiary structure, coming together to form a quaternary structure. One typical example of this is hemoglobin, where heme comes from the iron and globin is the protein part, and each of these globin chains, which are the tertiary structure of the protein, can come together to form a higher order of organization, which is referred to as the quaternary structure, to lead to the final function of the protein. So talking of function of proteins inside the cell, we first need to know how these proteins are synthesized within the cell. This is a subject which is quite detailed in the chapter of protein translation, but here I'm just going to give a brief introduction. So inside the cells you have DNA, which are converted into proteins through a messenger molecule which is called as mRNA. The DNA is formed by strings of nucleotides. It's a double-stranded molecule which is having these strings of nucleotides that are first read into an RNA molecule, and then this RNA is formed into chains of amino acids, basically forming the proteins. This reading happens through the protein synthesis machinery when each of the triplet of the nucleotides is converted into one amino acid. Now, what is shown here in the codon table below, that most of these amino acids are coded by more than one triplet codon, except for methionine here and for tryptophan, which is UGG. But most other amino acids are encoded by 4 codons, like for proline, threonine, alanine, as you can see here. Some of these amino acids, such as leucine, are coded by more than 4 codons, which is leucine and serine. Now, how do you get more than one codons being read into a given amino acid? So this is because of the property of the protein synthesis machinery, which is very specific to the first two nucleotides in its recognition, but is degenerate in its recognition of the third position. And that is where the variations in the third position of these codons are tolerated by the protein synthesis machinery, and it leads to the formation of same amino acid being linked. Another important point to note here is that of stop codons. So why these are called as stop codons? Because the protein synthesis machinery, when it recognizes these codons, it cannot incorporate an amino acid, and therefore the addition of the amino acids to a chain of a protein is stalled whenever this sequence is encountered. So in most cells you have 3 important stop codons, which is UAA, UAG, and UG. Yeah. So now that we have the basic introduction on the chemistry of the amino acids, as well as on the synthesis and the structure of the proteins, we move on to some of the analytical techniques that are employed in the laboratory to study these proteins. Now here, this is a table that gives you the purification profile of an hypothetical enzymes. Enzymes are nothing but proteins that have catalytic function. Whenever in a research laboratory you want to study a protein, you can either study it inside the cell or you can study it outside the cell. If you're interested in the chemical properties of the protein, such as its composition or its folding properties or its in vitro function, you would need to extract it out of the cell, purify it to the best possible purity, and then carry out your assays. So what is shown here is a typical table where you take a lot of cells. Let's say I'm purifying a protein or an enzyme from chickens. So I take tissue from the chicken. I buy it normally from the stores. I take it, I homogenize it so that the tissue is broken down in the cells, and then the contents of the cells are released. So that is the first step, which is that of crude cellular extract. Now I can measure what's the volume of the extract, and then I can say I have a method of measuring how much of protein I have in total in that extract. So I list that, and then within the total protein extract I have to find out the activity of my protein. In this case, since it's an enzyme, it has catalytic properties. Let's say it's an enzyme that can convert a simple disaccharide. A simple disaccharide is a sugar that has two sugar molecules into a simple uh, uh, monosaccharide. For example, lactose into glucose. So I do that reaction and I measure how much activity I have of total activity I have of that enzyme in the crude extract that I have got from the tissue of this particular animal. And then what I get is a specific activity, which is nothing but the total activity divided by the total protein. So it basically tells you how much activity you have per molecule of the protein that you have in your extract. But the first step is not enough. What you want is to have that particular enzyme in its purity, because right now you have just broken down the cell and you have a lot of other things present along with the enzyme. So what is typically done is a precipitation with ammonium sulfate. So this precipitation basically leads to precipitation of the protein fraction from the cell extract. So your protein gets settled down and all the other cellular debris are separated out. So now, because a lot of other junk material is gone, your volume is reduced, your total protein is reduced, the activity is reduced, but the specific activity increases because per unit of the because you have enriched for the protein fraction in your total cell extract. So since the denominator is decreasing and the specific activity is increasing. Now I could do further steps based on the property of my protein to get it to its most purest form. Now, if I know that the pI of my protein is very positive or it's very highly negative, I could do an ion exchange chromatography where I separate proteins with a certain kind of pI and all the other proteins are separated. This can be followed by a size exclusion chromatography, in which if I know what is the size of my protein, then I could separate it from all other proteins which are of different sizes. So the idea is that you combine the different properties of this protein to get it to its most purified form, and as you go on increasing the purity of the protein, you keep losing a lot of other proteins as well as your specific protein. So while you enrich for the specific activity because your specific protein has been enriched, you decrease the total protein amount and you increase the specific activity here, right? So how do you know that at each step whatever I intended to do, I am doing that? What you do what you do after this is to run each of these fractions onto what is called polyacrylamide gel. So this is nothing but a medium in which you put in your extracts. They are separated based on the different components of your extract are separated based on the sizes, which is what is shown in this figure. So if these are your different stages of purification, you can see that in the first step where you have the crude cellular extract, you have a lot of proteins which are represented by these different bands. Now during the second stage, when you precipitate it with ammonium sulfate, nothing much happens, but it's just the protein fraction of your extract that's get enriched. So in terms of the protein profile, that remains the same. the followed by a subsequent steps of specific protein enrichment, where you get given proteins enriched. So you can see that in this step, all the proteins that had certain isoelectric point or ionic properties were separated, and then proteins with a certain size, which is here, got separated, and ultimately you got protein that was specific for your interest. So this is an analytical technique which is routinely used in the labs to analyze the purity of your proteins. Another method which is a spectrophotometric based method you could use here is that you can look at how much of light is absorbed or emitted from the solution of the proteins. So because of as I told you earlier, that each of these amino acids are different in their chemical properties. Because of the differences in their chemical properties, they also have differences at the wavelengths of light that they either absorb or emit. So tryptophan and tyrosine are typical R groups that absorb at 280 nanometers of wavelength. You have other amino acids most of the other amino acids do not really have very high absorption, except for positively charged amino acids that can give you a peak at 230. The peptide bond can also be a signature that could be used in certain spectrophotometric assays, but it's relatively weak. So here, using this property of absorption of these amino acids that are usually present in most proteins, you could calculate the total amount of the protein that is present in your solution using this relationship, which is called as Lambda Beard Law, based on the name comes from the name of people who made this law, where you have the intensity of the incident light and the intensity of the transmitted light. The ratio of that is directly proportional to the concentration, which is C here, the concentration of the protein. And then you have constants, which is epsilon, which is specific to a given protein, and L is the length of the cuvette in which these measurements are being made. So essentially you see that the ratio of your incident light to the transmitted light is directly proportional to the concentration of the protein. So greater the concentration of your protein, greater is going to be this ratio. So if through our experiments we know this ratio, we can indirectly calculate the concentration of our protein. Okay. So now I have some questions here which would probably help in clarifying some of the concepts that we reviewed in the earlier slides. So the first question here is, in a globular protein, hydrophobic amino acids are generally found on the surface, on the interior, both on the surface and the interior, in the hinge region of the molecule. So hydrophobic amino acids are those amino acids which due to their chemistry or due to the R group basically which they have, they repel water molecules. They are hydrophobic. So these are non-polar, uncharged or aromatic amino acids. Now because of their property that they repel water molecule, they obviously are most likely to be present in the interior of the molecule rather than being on the surface, where they have higher chance of being in contact with water. Second question, for performing SDS gel electrophoresis, an excess of beta-mercaptoethanol is added to protein samples for the purpose of. So this might look like a memory question, but if I tell you the structure of beta mercaptoethanol, you would be able to know what it does to the protein. So beta mercaptoethanol, ethanol, as you know, is CH3CH2OH. If I have to add, so this is my alpha carbon, this is my beta carbon. If I have to put a mercapto group, it's going to be an SH group, and a mercapto group is an SH group. So now if now that you have the beta mercaptoethanol structure, you know what exactly would it be doing to the proteins. Next question, a 20-year-old anemic man is found to have an abnormal form of beta globin that is 172 amino acid long rather than a 141 that is found in normal proteins. Which of the following point mutations is likely to be associated with this abnormality? So as you can see here, what's mentioned is that under usual circumstances, this protein has 141 amino acids, but in this particular case, it has 172 amino acids, which means it has more amino acids that it is supposed to have, which means that where the protein was being synthesized in the cells, it did not stop at a point it was supposed to stop, and therefore more amino acid got incorporated into the peptide chain. So now that you know that there is a problem at the stopping of the synthesis of the amino acid, there must be a difference, or there must be a mutation at the stop codon. We know that there are 3 stop codons usually present in most cells. So there has to be a change in these to a coding amino acid for this abnormality to come up. Next question. Which of the following is responsible for degeneracy of a genetic code? This is a straightforward information question, and we've discussed this in the next question. How many decapeptide variants will result if during peptide synthesis 10 of the 20 amino acids are allowed to be incorporated randomly? So here we are talking about decapeptide. Decapeptide is a peptide having 10 amino acids, and for each of the 10 amino acids you could substitute 10 out of the 20 different possibilities. So if at each position I have 10 different possibilities available, you can calculate how many random variants could be possible. Next question. Biosynthesis of protein employs which of the following precursors? So to answer this, you need to look at the structure of the protein and structure of other amino acids, and you would very easily be able to find out that what could be the possible precursors from looking at the number of carbon atoms or the identity of the atoms that you have in the side chain for the 2 different amino acids. Next. Which of the following amino acid is found both in D and L forms in peptidoglycan? This is an information question, but it's interesting in the sense that the biologically relevant amino acids are usually present in their L forms, but in certain structures, such as in peptidoglycans, you have the D amino acid incorporated. These are most commonly found in cell walls, and here this giveaway answer. The DNL form of alanine are usually present in the peptidoglycan or membranes. Okay, thank you.